Base excision repair in eukaryotes. With base excision repair being present in all living cells, right from prokaryotes to higher mammalian systems, it would be interesting to note what is the molecular mechanistics of base excision repair in eukaryotes. So let us look at the learning outcomes of this session. Base excision repair addresses lesions in DNA that result in little or no distortion at all in the double helix. These lesions are generally as a result of oxidation, alkylation, spontaneous hydrolysis, deamination, etc. DNA glycosylases are the enzymes that are responsible for recognizing these specific DNA lesions. And so it is observed that every DNA lesion has its specific or a group of DNA lesions falling under the same umbrella would have a specific DNA glycosylase that would not just recognize it but also remove the damaged base. The removal of the damaged base will lead to formation of what is called as an abasic site. And these abasic sites are then repaired by molecules that enable either short patch repair or long patch repair. So BER itself can have two types. One is what is called as a short patch repair and the other one is what is called as a long patch BER. BER, of course, therefore minimizes the manifestations of these damages that can lead to cancer, aging, and other neurodegenerative diseases. Getting an overview of what are the damages that are taken care of by BER. So you have what is called as oxidation, damages that are happening in the bases due to oxidation. So as you can see over here, 8-oxoguanine is one common oxidation damage that happens. So you can see that at the C7, you would have the oxidation taking place, forming what is called as 8-oxo-G. You can also have 5-hydroxycytosine being formed due to oxidation. You can also have thymine glycol formed due to oxidation. You can have FAPYG that is forming because of oxidation. So all these are results of oxidation. You could also have under the BER supervision or repair, you could have spontaneous deamination that happens. For example, guanine getting deaminated to forming hypoxanthin, or cytosine, where it had an NH2 group over here, is deaminated. In place of that, you have the double bond O formed, and therefore this becomes uracil. And we all know that uracil is an unnatural base in the DNA molecule, and hence it is removed by the BER system. So alkylations as well as oxidations as well as spontaneous hydrolysis. So for example, you can see over here there is a spontaneous hydrolysis making the sugar an aldehyde, or you can also have over here, as you can see, a hydrolysis leading to, or you can have removal of a base itself. You can see that the base has been removed to form an basic site. So these are all DNA lesions that are formed due to oxidation, alkylation, spontaneous hydrolysis, deamination, etc. Now, these damaged bases, they do not really create any distortion in the double-stranded DNA, but what can happen is they are able to not base pair correctly. They will base pair with a wrong nucleotide. So for example, 8-oxoguanine can form two hydrogen bonds with cytosine, but it can also bind to adenine. So therefore here what happens is that the base pairing changes. Similarly, uracil, instead of cytosine, cytosine would bind to guanine, but now because of the presence of uracil, you would have it binding to A. So therefore, the base pairing changes, and these single point mutations have led to a lot of cancers, right from epithelial cancers to mucosal cancers, etc. So therefore, if these are not corrected, then it can be very detrimental to the cell, and not just to the cell, to the entire organism. So therefore, BER has a very important role to play in the eukaryotic system. Now let us look at the fundamental steps in the BER pathway. So the first step is recognition and then excision of the damaged nucleotide, and that is taken care of by DNA glycosylases. Then it is of course the removal of the sugar phosphate at the A basic site, which is by an enzyme that is called as AP endonuclease or exonucleases. And what has been observed is many a times the incision in sugar phosphate backbone can also be created by some DNA glycosylases. Once that has been done, then because it becomes a gap due to the removal of the sugar phosphate backbone, it has to be filled up, and that filling up is done by a DNA polymerase. And then, of course, the sealing is done. The nick is sealed by DNA ligase. So, therefore, these are the three basic steps which was proposed by Thomas Lindahl in 1974 as he studied one of the DNA glycosylases and the base excision repair thereof. Now, BER takes place both in the nuclei of cells as well as in the mitochondria. The mitochondria also having DNA, mitochondrial DNA replicating, mitochondrial DNA also going through various oxidations, alkylations, etc. So, damages in the mitochondrial DNA are also taken care of by the base excision repair mechanism. Now, the first step is, of course, in the first step is the base removal. So there are 11 distinct DNA glycosylases that are present that can remove the base. And just to get a glimpse of these 11 distinct DNA glycosylases that are present in mammals, in plants, etc., one type or the other are present. 11 distinct are definitely found in the mammalian systems. So one of them is uracil and glycosylases. This basically takes care of presence of uracil, which is originating because of deamination of cytosine. And this enzyme is very highly specific for uracil removal itself. Then you have single strand specific monofunctional uracil DNA glycosylase 1, another enzyme that is responsible for removal of uracil. You have the oxojoy DNA glycosylase. So this is specifically a glycosylase that is taking care of the damage that happens to guanine because of oxidation. You have thymine DNA glycosylase. This has as its substrate range several substrates, including thymine, 5-fluorouracil, etc. You have methyl binding domain glycosylases. So this is another glycosylase that takes care of damage that happens because of methylation. You have methylpurine glycosylases itself. So say for example, you have the formation of methyl guanine or you have the formation of methyl adenine, then you have this specific glycosylase that takes care of removing these methylpurines. You have what is called as mute Y homolog glycosylases. You have what is called as NTHL1 endonuclease 3 like glycosylases, and you of course have three distinct glycosylases which are endonuclease 8 like glycolysis. Now, what is interesting to note is that all these 11 different distinct DNA glycosylases fall majorly under four superfamilies. So the four superfamilies include uracil DNA glycosylases. So whether you have UNG or whether you have SMUG, they fall within the uracil DNA glycosylases. And this classification or this falling within the uracil DNA glycosylases is because of the structural and functional similarity that is there between the glycosylases. Then you have what is called as helix hairpin helix glycosylases. This is a typical motif that is present in the glycosylases, and any of the glycosylases that have this typical motif fall under this superfamily. You have endonuclease 8 like glycosylases. Again, this is basically looking at the fact that they are sharing certain structural aspects and of course therefore the functional aspects. You have what is called as the methylpurine glycosylases. So these are the four superfamilies within which you have the 11 distinct DNA glycosylases present. But What is noteworthy over here is that certain set of substrates are looked after by a certain DNA glycosylases. In other words, the DNA glycosylases have their own specific substrates. Now, how does the DNA glycosylase remove the base? If it has to remove the base, it has to identify or detect the base. So therefore, in order to find out which base it has to remove, it will obviously have to scan the genome. And what has been observed is that uh, different DNA glycosylases have different mechanisms by which they scan the genome to identify the damaged base. So we here are going to just take 2 or 3 examples of DNA glycosylases to see how they scan the genome and are able to identify and detect and verify that it is the base that it has to basically remove. So let's look at first mute Y homolog glycosylases. What is interesting to note over here is that the mute Y homolog glycosylases within their structure have about 4 FES clusters, and these 4 FES clusters exist in a particular oxidation state. So they exist as 2 plus oxidation state, but they can go to 3 plus oxidation state, that is a higher oxidation state. So it is because of this principle that it can go from one oxidation state to another oxidation state by taking up an electron or giving an electron. This particular principle is used by the mute Y homolog glycosylase to actually identify the base that has to be removed. So what is observed is that mute Y homolog glycosylases move in duplexes. That means they move two together on the DNA. So therefore they would have a DNA binding domain. They would have a motif that will help it interact with the DNA, and two mute Y homologs will be present adjacent to each other and then move on the DNA. Now, as they move, they keep changing the FES state from one oxidation to state to the another oxidation state, and there is an exchange between two glycosylases. This exchange is through the DNA. So, therefore, the transfer of the charge to the neighboring glycosylase is through the DNA. So therefore, what is observed is that if the DNA is normal, that is, there is no wrong nucleotide or modified nucleotide or oxidized or alkylated nucleotide present, then there is a continuous exchange of electrons between the glycosylase and the DNA, and that is how the glycosylase moves through with the DNA. But when you have a damaged base present, then what is observed is that the glycosylase halts over there because there is no smooth transition of electrons from the DNA to the glycosylase. And therefore, what happens is one of the glycosylase that doesn't change the oxidation state gets removed from the DNA, while the other one remains in contact with the DNA at the lesion. And so that is how the mute Y homolog glycosylase identifies the base lesion. Now, of course, there is a lot more to understand with respect to how there is a transfer of electrons from the DNA to the glycosylase and back to the DNA so that the charge distribution is maintained all through. But this is what has been interesting to note, and the study has been very interesting. Another example that is there is of OGG1, that is oxoguanine glycosylase 1, and UNG, that is uracil and glycosylase. Both these enzymes have at its mouth, so it has a pocket with an active site, and at the mouth of the pocket are present certain specific amino acid residues, and these specific acid residues are able to interact with the chemical surfaces of the specific modified bases. That is, if it is oxidized, okay, then the residues present in the mouth and the mouth of the active site is able to interact specifically with that oxidized base. So therefore, what happens is that the mouth of the uh, active site of the OGG1 and the UNG are able to discern a normal base from an abnormal base. And that is how it basically identifies where exactly the base lesion is. What is also interesting to note is that both OGG1 and UNG are existing, or they interact with the DNA as an open complex, but when it interacts specifically with the modified base, then it forms a more closed complex. So therefore, the conformation goes from a loosely bound complex to a more closed complex, and this change in the conformation from an open, loosely bound enzyme to a more closed conformation marks the presence of the base lesion. So if you have normal bases, then the enzymes remain as open complexes, but they are attached to the DNA and they are scanning the DNA. But when it reaches a base lesion, then from an open conformation, it becomes a closed conformation, and that marks the presence of the lesion. So therefore, this is what is called as a pre-selection of damage. Now, what do we mean by pre-selection of damage? See, we all know that DNA glycosylases are known to flip out bases into their active site to remove the base. But before flipping out, these enzymes are able to actually identify the damage, and therefore it is called as pre-selection of damage. Now what happens next is, of course, the DNA glycosylases have to verify that it is a damaged base. So how does it verify? It generally verifies by rotating the base, and on rotating the base is flipped out into the pocket, and once it is inside the pocket, the active site has very specific microenvironment that can interact with the flipped base to carry out catalysis and break the glycosidic linkage between the sugar and the base. Thereby, after verification, so if the base is not fitting into the active site or it is excluded from the active site, then of course catalysis will not happen. So only after verifying it, the active site getting induced fit into the damaged base, only then catalysis will happen, otherwise catalysis will not happen. So if there are non-specific interaction, then catalysis will not happen. But if there are specific interactions, then catalysis will happen. So that is a mode of verification of the damage and then removing the base. So we again take the example of UNG, that is uracil and glycosylase. So the uracil and glycosylase, on its outer surface of the active site, okay, they have amino acid residues that are able to interact with the minor groove of the DNA. And in that outer surface space itself, they have what is called as a hydrophobic chain with a conserved leucine, and that is responsible for flipping the base. You can see the base was here, now it is flipped in, and this is the crystal structure of the UNG present. So you can see how the base is flipped in. So you can see the flipped in base over here. Okay, that is thanks to a conserved leucine. And as the UNG attaches itself to the minor groove of the DNA, it forms a 45 degrees bend. So that is enhancing the flipping out of the base into the active site of the UNG. So with this, what happens is the purines, these are, uracil is very specifically able to fit into the active site. So if there are purines present at this site, so if the base is not a uracil and it is a purine, then the active site or the pocket is so narrow that it will not allow the purines to enter, and therefore they are sterically excluded. So you can see how it is maintaining its specificity by ensuring exclusion of a nucleotide which is double ringed rather than single ring. But then the question arises is that there are other pyrimidines present. So for example, if you have a thymine, thymine is very similar in structure to the uracil. So in that case, even if it is a narrow pocket, how will it prevent the other pyrimidines to enter the active site? So what has been observed is that they have another side chain which has a conserved tyrosine that can interact with the thymine and other methyl pyrimidines not allowing them to enter the active site, but only uracil is present inside the active site. So you can see how the UNG is able to maintain its specificity, and on binding to the specific uracil, it is verifying that the damaged DNA is uracil, and that it will remove the uracil by breaking the glycosidic linkage. Now, another interesting facet about the DNA glycosylases is that some DNA glycosylases are monofunctional and some DNA glycosylases are bifunctional. That means the word itself is indicating that this is having a single function, whereas this has two functions. So let us understand what is the difference between monofunctional glycosylases and bifunctional glycosylases. So here we take the example of uracil and glycosylase and thymine DNA glycosylase. They both fall under the monofunctional glycosylases, while the oxo guanine glycosylase 1 falls under the bifunctional glycosylase. As mentioned earlier, you can see that UNG is existing as an open conformation, but when it reaches the base lesion, it gets converted into a closed complex with the U getting flipped into the pocket. Okay, so you can see that it is flipped into the pocket, and within the environment is present water molecules that act as weak nucleophiles, so that using this water as a nucleophile, it is able to break the glycosidic linkage. And with the break of the glycosidic linkage, the uracil is removed and it goes away, and you can see that of course the active site in the UNG is the aspartate, and the aspartate is able to interact closely with the opposite nucleotide, also ensuring that it is reading the base pair and not just the uracil, but that it is able to cleave off the uracil from the AUA base pairing, and considering that it is able to recognize that it is a wrong nucleotide and it can remove. So therefore, monofunctional is just indicating that this glycosylase has only one function, and that is cleaving of the glycosidic linkage between the base and the sugar. So here what happens is you are having now an A basic site present, okay, with even the phosphate sugar backbone intact, only the base being removed. Now let us go to thymine DNA glycosylase. So the thymine DNA glycosylase also exists as an open conformation, but when it binds to the DNA, okay, it closes in, okay, with its N-terminal end having specific recognition for thymine, okay. And interestingly, what is observed is that the thymine DNA glycosylase is able to recognize a T that is bound to G but not to A, which means that within the microenvironment there are residues that are also able to look at the opposite base that is there with the T. So it does not just look at T alone. So T definitely in the DNA is a normal base, but when T is attached to a wrong nucleotide, then that T has to be removed. So how does the thymine DNA glycosylase realize that this is not the correct T present at the correct site? So therefore, it looks at the base pairing. If the T is with the A, then there is no problem. But if T is bound to some other nucleotide which is not complementary, then that T has to be removed. So therefore, you have several residues present within the microenvironment of the pocket that is not able to just interact with thymine, but it is also able to interact with the opposite base, and that interaction enables it to recognize that T is bound to G and not to A. So that conformation that is there because of the TG and not because of the TA is also something that is very specifically observed by the thymine DNA glycosylases. So the aspartate that is, again, the asparagine that is present over here, it is noted to be able to interact through hydrogen bonding with the G, or it is able to ensure that it is linking to the thymine, and then of course you have the glycosidic linkage between the thymine and the G breaking up, sorry, the thymine and the sugar breaking up, so thymine is released, okay, and you have an A basic site formed over here. Now, in case of thymine DNA glycosylase, what is observed is once it forms a closed complex, it remains bound to the A basic site as a closed complex. In fact, what has been observed that to a A basic site, the glycosylase binds with higher affinity. And so, in order to remove the thymine DNA glycosylase from the A basic site, to allow further processing of the repair system, a post-translational modification has to be carried out on the thymidine DNA glycosylase to change its conformation from closed conformation to open conformation, and only then you have the TDG being released. So that over here has been observed to be done by what is called as sumoylation. Okay, so this is important for many glycosylases because when they interact with the A basic site, their interaction is observed to be stronger. So to change that conformation from closed to open, a certain post translational modification is needed to be done. Now we go to the bifunctional glycosylase, under which falls the OGG1. So you can see that the OGG1 also is existing as an open complex, and it closes slightly when it comes to the OXOG. Now, it has a lysine as the conserved amino acid residue within the pocket, and through the specific interaction between the OXOG and the OGG1, it is able to form a glycosidic linkage and release the OXOG. But what it also does is it is able to form a nick in the sugar phosphate backbone because of the presence of a lyase activity. So you can see that it has two enzymatic actions. One is breaking the glycosidic linkage between the base and the sugar, and the second is creating a cut in the sugar and the phosphate in order to form a nick. And therefore, you have now not just an A basic site, but you have a nick at the A basic site as well. In case of the OGG also, to remove it, a modification has to be done in its conformation so that it releases from the A basic site. So this is something that is extremely important, that any bifunctional glycosylase apart from its glycosylase activity also has a lyase activity present. Now we go forward that after the job of the DNA glycosylase is done, what happens next? So let's consider that this is the base which has either oxidation or alkylation or deamination. So this is the base that has been damaged. So it's the lesion, base lesion, and then DNA glycosylase removes the base. Okay. So after it removes the base, you have a basic site. So a basic site, which means you don't have the base present over here, but you have the ribose with the sugar. You have the ribose with the phosphate link on both the sides. That is at the 3' end as well as at the 5' end. Now, from here onwards, there can be three possibilities. Okay, so you can have, if the DNA glycosylase is monofunctional, okay, then what comes in is another enzyme called the AP endonuclease 1. AP stands here for apyrimidinic or apurinic. So basically, it is an enzyme that is able to recognize an A basic site. So it comes and binds, especially when the DNA glycosylase is interacting, and sometimes on binding of the APE1, the DNA glycosylase leaves. So when you have the APE1 coming in, the APE1 is able to cut the bond between the phosphate at the 5' end of the A basic. So if this is the A basic site, at the 5' end of the A basic site, you have the cut happening over here, releasing what is called as the 5' deoxyribophosphate and leaving a 3' OH over here. Okay, this is one possibility. The second possibility is that if DNA glycosylase is bifunctional, if that is the case, then after the removal of the base through beta elimination, acting as an AP lyase, you can see that at the 3' end you have a cut being formed, okay, and that cut is enabling formation of an unsaturated hydroxyaldehyde. So this unsaturated hydroxyaldehyde, okay, that is formed is the 3 prime. It is equivalent to the 3 prime deoxyribose phosphate, correct? So in this APE1, you have the 5 prime deoxyribose phosphate being formed, while in the case of AP lyase, you have the 3 prime equivalent 3 prime deoxyribose phosphate being formed, leaving a 5 prime phosphate free in this case. Now, in some cases where DNA glycosylase has a bifunctional role, it not just carries out beta elimination, but it can also carry out the delta elimination. What does that mean? It means that the sugar component can be cleaved at this site as well as at this site. So you can have beta elimination, you can also have delta elimination. So here is beta elimination, here is delta elimination. So what is released is an trans 4 hydroxy 2,4 pentadienal. So the ribose is removed in this form, okay, and what it leaves behind is at the 3 prime end a phosphate group and at the 5 prime end also a phosphate group. So this is what happens next after the DNA glycosylases come and do. So in all these cases you have an A basic site with a nick that has been formed with respect to the shock sugar and the phosphate of the DNA. Now we look forward. So you have in BER a short patch BER. So we will understand what is a short patch BER. So once you have the APE1 forming the 5 prime deoxyribose phosphate or the AP lyase forming equivalent to the 3 prime deoxyribose phosphate, or you have the ribose entirely removed and 2 phosphates remaining, what can now happen is that you have to ensure that one end has OH, so that becomes 3 prime OH, and the other end has 5 prime phosphate. So when you have a final nick, the nick should lead to the phosphodiester breaking in such a way that you have the OH and you have the 5 prime phosphate present. So in case of the AP1 mediated cleavage of the sugar phosphate, you will find that the Pol beta has inherently, other than its polymerase activity, present what is called as a phosphodiesterase activity. So because of the presence of the phosphodiesterase activity, it can remove the 5 prime uh, deoxyribose phosphate. So you can see this entire part is going to be removed, okay? And because this entire part is removed, what remains is the 3 prime OH and the 5 prime phosphate over here. Now, in case where the DNA glycosylase was able to carry out beta elimination and leave an equivalent of 3 prime deoxyribose ribose phosphate present, the APE1 will now come and cleave at this site. Okay, so it will cut at 5 prime of the A basic site to form again a 3 prime OH and a 5 prime phosphate end. So that is what is happening. Now, in case where the DNA glycosylase acted both or was able to carry out beta elimination as well as delta elimination, leaving two phosphates present, 3 prime phosphate as well as 5 prime phosphate, this phosphate has to be removed. And so you have another enzyme coming in called as the polynucleotide kinase phosphatase. So it's a bifunctional enzyme. It can act as a kinase as well as act as a phosphatase. And this enzyme removes the 3 prime phosphate, leaving a 3 prime OH. Okay. So in all eventualities, what you have is now after the end processing, you have a 3 prime OH remaining at the A basic site and the 5 prime phosphate remaining at the A basic site. What happens next? The DNA polymerase beta is able to associate with XRCC1. This XRCC1 is a X-ray repair complementing protein 1 and it is involved in several DNA repair mechanisms. The Pol beta adds the nucleotide. So you can see it adds a nucleotide based on what is present in the template. Okay. So it will add the correct nucleotide. Okay. And then after adding the correct nucleotide, DNA ligase 1 or DNA ligase 3 with XRCC1 comes and seals. So when you have only one nucleotide, being repaired, then it is what is called as a short patch BER. That means the displacement is only of one nucleotide and not of several nucleotides, and so it is called as a short patch BER. Now we go to the long patch BER. Now in the long patch BER, as you can see, this is the APE1 cut A basic site where you have the 5' deoxyribose phosphate present. So what happens is that using this double strand that is present over here, RFC is going to load PCNA. And this PCNA is going to bind to the pol beta or to the pol delta epsilon, any of the polymerase, and it is going to introduce new nucleotides by displacing the existing strand. So you can see it has what is called as the strand displacement activity. So therefore, since it has since it has displaced the existing strand that is present, this forms a flap. And when there is a formation of flap, the PCNA is able to interact with FEN1, which is an endonuclease, and that FEN1 is able to remove the flap, right? So what again remains eventually is with the removal of the flap or the nucleotides that have been displaced, the strand that has been displaced, you have the newly synthesized sequence present over here and the final nucleotide which has to be just sealed. So this sealing is again done by DNA ligase 1 and therefore it completes what is called as a long patch BER. So this is how you have the base excision repair carrying out its repair. And interesting to note is that you would always have the PCNA enabling, and we all know that PCNA having a trimeric structure is able to bind to several proteins and maneuver not just replication but also help in the DNA repair mechanisms. So it is PCNA that not just binds to FEN1, it binds to the DNA polymerases, it also binds to the DNA ligase 1, forming that complex to ensure that you have the entire long patch BER complete. So with this, let us come to the concluding remarks. The DNA lesions due to alkylation, oxidation, etc. are recognized by specific DNA glycosylases due to its ability to scan the DNA, and as it reaches a lesion, it binds strongly. These damaged bases are verified by flipping the base into the active site of the enzyme for catalysis of breaking the glycosidic linkage between the base and the ribose, leaving an A basic site. These A basic sites are then subject to incision either by APE1 or if the glycosylase also has a lyase activity, then through the lyase activity, it will carry out the incision at the A basic site. And these are then followed by mechanistics of either short patch BER or long patch BER. The DNA polymerase beta is responsible for strand displacement and synthesis in the BER, which in turn are aided by PCNA FEN1 DNA ligase. Mutants of DNA polymerase B has been associated with several epithelial cancers, and therefore it has been very importantly noted that DNA polymerase beta, which belongs to the X family of DNA polymerases, has a very important role to play in base excision repair. Thus, BER is a molecular mechanism that sequentially enables repairing DNA damages that, if not repaired, can lead to permanent mutations, which in turn can lead to aging or diseases like cancer. Thank you.